So here are our samples from today. Today we only sampled from 2 turtles, so this is the perfect amount of turtles to show you what we do for our lab work. First off, I'm going to be carrying the samples and bringing them over to the ESR station.
Here I am working at the ESR station, or the erythrocyte sedimentation rate. This is my summer research, and the first thing that I'm doing is inverting all the blood tubes so that they can mix. Then I am taking the microhematocrit tubes and filling them 3/4 of the way up with blood, plugging the tubes with the clay, and sitting them upright for 1 hour. ESR has been known to be an indicator of chronic inflammation in other animals, but it is just recently being studied in chelonians. So that's why it's really important that we look at it in the blandings this summer. After ESR, the second thing we do when we get back in the lab is start spinning down the plasma. So the first thing you can see me doing here is changing all the settings on the centrifuge. We want to make sure that for plasma, it is going to be set at 6,000 RPMs and running for 10 minutes. Later, you'll see us change these settings again when we're ready to spin down the capillary tubes. Now I can put the blood into the centrifuge. As I do that, I want to make sure that each tube is sitting across from another so that they are balanced when it spins down.
So using the same microliter container of whole blood, I'm going to start making blood smears. My first step, like ESR, is to invert the blood tubes to make sure the blood is thoroughly mixed together. And then I'm going to attempt to make my slides. What's very important here is that I'm not actually hitting the table because that would disrupt the ESRs that are also on the same workstation, but I am tapping my other hand so that the pressure is on my hand and there's no movement at the table. Here you can see I've put 1 drop of blood onto the slide, and with medium pressure I pull it back, make sure it spreads across, and then push forward. You'll get a close-up of that soon, but it's standard procedure for us to make 2 blood smears per every turtle that we get, because eventually we're going to be reading cells on these blood smears, and in case something happens to one, we always like to have it back up. I like to hold my blood smears up to the light to make sure I've created a feathered edge. Here you can kind of see that feathered edge and the chrome color it gets when it's put up to light. That way you can make sure you have a monolayer so you can visualize your cells. Here what I'm doing is I'm noting what date the sample was taken on and also the sample number of the turtle. So each turtle is given its unique number, and the unique number is then associated with the blood smear from that specific turtle. Using the same blood from turtle 1, I'm filling a capillary tube about 3/4 of the way up with blood. These tubes are then going to be plugged with clay, and they're going to be used to run a PCV-TP, and we will explain what all of those letters mean when we get to it. But just like making 2 blood smears, we make 2 of these tubes just to make sure that the numbers line up. So now it's time to do the blood smears for the second turtle, and this is just going to be the close-up version of what I did before, so that way you can see me putting the 1 drop of blood onto the side, and you can see my technique of pulling back, letting the blood spread out, and then pushing forward.
Next, we're going to set up the hemocytometer station. First thing we need to do is pull out the microscope from its protective bag and plug it in.
So the first thing I like to do is grab a piece of paper or a paper towel and write down the sample numbers, and I like to have this piece just to make sure I have a place to record what times I'm doing what. So first I invert the tube several times to mix the blood. This is still using the same whole blood microtainer that we've been using for everything else. And you see me take a bit of the blood and start to mix it in with the dye for about 30 seconds. Here what we're doing is we're letting the blood cells sit in this specific type of pink dye for a certain amount of time. In this case, it's 10 minutes, so that way the blood cells in the blood will stain the bright pink color. Since we have 2 turtles today, you can see me then doing this for the next turtle. The pipettes are pre-measured, so that way we don't have to measure out the blood every time we do it. The pipette will always grab the perfect amount of blood for these individual dye containers. After I place on the lids, I then grab my pen and write down the time that I put the blood into the dye. Now I let it sit for 10 minutes. In this time, I also like to set up my hemocytometers so that way when the 10 minutes has passed, I'm ready to load them. Usually we do 2 hemocytometers per 1 sample of blood in case one messes up along the way, but since we only have 2 turtles today and we have 6 hemocytometers, here you can see me setting up 3. This is what a hemocytometer looks like close up. It is glass on either side, and then it contains 2 chambers with grids on them per hemocytometer. After we place the hemocytometer down, the next step is to place the cover slip on top, and then these 4 right here are ready to have blood put inside. So here you're going to see me finishing setting up the last 2 before we load the blood.
Right now, while ESR is running, plasma is spinning down, and the blood is sitting in the dye for the hemocytometer, we take this time to write in our lab book the date and the name and sample number for each turtle. This notebook is the place that we immediately write down all of our data as we finish each lab test.
So finally, after 10 minutes, the plasma is done spinning down. The centrifuge is used for multiple tests, so it has different accessories to fit inside of it. Right now you can see me pulling out the accessory that we use for plasma. What we do is we gently tip it back and we can grab the samples. Now I'm going to show you what it looks like all separated. When I show it to the camera, you can see in the top there's yellow plasma, and in the bottom there's a pellet of blood, and in the middle is the separator. So what we do next is we uncap it. We open an Eppendorf tube, struggle to open an Eppendorf tube apparently, and then we just pour it directly into the tube. These samples will be frozen so that way we can use them to aliquot plasma in the future when we return back to the lab down at the university. I do this for both samples, so then I unplug the next one, gently pour it into the Eppendorf tube, making sure to get every last drop out. This is like liquid gold. And then we can move on to our next step in our lab work.
Now you can see me replacing the blood tube holder for a different accessory that will be able to hold the capillary tubes. Because the whole point of a centrifuge is that it is spinning very quickly, it's really important that we make sure that each component is screwed in tightly. Now I am once again changing the settings back down to 5,000 RPM and running for 5 minutes. Now that we're all set up, I can go grab all of the capillary tubes that Sam already filled with blood and carefully carry them over. Because the capillary tubes are too small to write on themselves, both the clay stand that they sit in and the accessory in the centrifuge are numbered. Since the centrifuge still has to be balanced, each sample sits across from itself. You can see here that sample 1 and 13 are from the same turtle, and then samples 2 and 14 are also from the same turtle. The last thing that I do before I put the lid on is push up against each capillary tube to make sure that they are pressed against the side. This will hopefully decrease the chances of them breaking while they're spinning down. Now that the blood has sat in the dye for long enough, it is time to fill the hemocytometers. I do this by carefully pipetting up the blood and dye mixture and then filling both chambers on each hemocytometer. As Sam mentioned, in this case, we're using 3 hemocytometers per turtle sample. So I will repeat this process 3 times with the first sample of blood, Before I move on to the next sample, I am once again going to write down the time that we put the blood and dye mixture into the hemocytometers. They need to sit for another 10 minutes before we can read them. Now we're ready to move on to the next sample. Here's a close-up view of me filling each chamber. While we are once again waiting on the hemocytometers, I can move back over to the centrifuge to get out all of my capillary tubes. Now that the blood has spun down, you can see 3 layers. At the top, there's plasma. In the middle, which is hard to visualize, is a buffy coat, and then at the bottom is all the cells. I carefully add all the capillary tubes back into their holder and move them over to the table where I can do a PCV and a TP. PCV stands for packed cell volume. So right now you can see that I am lining up the top of the plasma with the top line on this chart and the bottom of all the cells with the bottom line on the chart. Right where the plasma and the cells meet is the PCV. You can see I am going to repeat this process with the second capillary tube from the first sample to make sure that both tubes from the same sample agree. Once I have the PCV, I can move on to the TP. TP stands for total protein. TP is read on a refractometer, so the first thing that I need to do is take the capillary tube and carefully break it in half near where the plasma and the red blood cells meet. I then can tap a little bit of the plasma onto the refractometer and look into a light where I can read the value. Before I repeat this step, first with the second capillary tube from our first turtle, I will clean the refractometer with a Kimtech wipe. After reading the TP on the second capillary tube from the first turtle, I will write down all the values and repeat the packed cell volume and the total protein for the second turtle.
Here is the view we see in the microscope from a hemocytometer. You can see here the 9 grids, and then now you can see a zoomed in version of what the grids look like to us when we count the cells. In mammalian species, an analyzer can be used to count the blood cells, but for our chelonians, we have to count them by hand using a single cell counter. So here you can see me scrolling through the grids, and every time I see a hot pink cell, I click my thumb on the single cell counter. We have to make sure that we're following the same protocol the entire time we do the hemocytometers with every hemocytometer we do, so that way our results are accurate. So I read from top to bottom and from left to right in a zigzagging formation through my hemocytometer. I have now finished reading half of the hemocytometer, and I got 101 cells for the first side. Now I begin to click the second side. And here I'm very sad because I realized that they are not within the percentiles they need to be. One side was severely different from the other side. So that, ladies and gentlemen, is why we make more than one. So instead of having to load and wait another 10 minutes, I already have a second hemocytometer prepared for that sample. So here I go again, round 2. After counting again, the first side of this one ends up being 88. Now on to the second side. And ta-da! The second side was 91, so these two are within 90% of each other, so they are accurate. Now you see us cleaning up the hemocytometers after we're done using them. We use Kimtech wipes to wipe off the dye that's directly on the cover slips and the slides, but we also use rubbing alcohol to deep clean the hemocytometers almost every day that we use them. So here you can see Kayla removing the cover slips, rubbing off the dye, and then placing them back in their protective containers.
Now that 60 minutes have passed, I am ready to read the ESR. Here you can see me using calipers to measure the distance that the erythrocytes have fallen from the top of the plasma to the top of the cells. And you have a better view of that right here. After I read both samples, I will also write this down in our lab notebook. Now that the slides from the blood smears are all dry, they are ready to be loaded up into a slide holder so that I can stain them all at one time. Before we can start staining, the first thing I need to do is set up our station. Here you can see 3 different containers that will each hold a liquid from the 3 different steps of dyeing. The first that I'm pouring in right now is a fixative. Next, I will pour in the first dye and then finally the second dye. To show you just how realistic this video is, here you can see me accidentally pouring the second dye all over our counter, and I later have to clean that up. Next, you can see me turning on the water and making sure that it is not running too forcefully, and then beginning to dip the slides into each container. After dipping the slides in the fixative, I move on to the first dye and then finally the second. Then I carefully rinse off all the dye by holding the slides on the sides so that the water does not directly run onto the blood smear.
Here you can see me putting away the slides that were dyed from yesterday. The reason why we dye slides is to perform white blood cell counts. So it will be my job when I return back from school to count 100 cells per slide of each turtle and then report what the counts are for each type of white blood cell. Turtles have lymphocytes, monocytes, heterophils, basophils, and eosinophils. For some reason, the Blandings specifically are very lymphocyte-heavy turtles, so those numbers are a little bit elevated in particular species. We look at white blood cell count to look for health status of individual turtles as well as the overall population. If certain turtles have either higher or lower white blood cells, we can try and determine whether there is an underlying infection or disease that is occurring within the population or that specific turtle.
Then I bring the slides back into our lab so that they can lean against the wall and dry until the next day, when they will also be put away. After we're done staining all the slides for that day, we start cleaning up the staining station. I do this by pouring each dye back into its original bottle. It's important we do this at the end of the night because otherwise the stains will evaporate. One thing we did not include in this video is that Sam and I, at the end of the day, will spray down all of our counters and common spaces with Knoll sand or bleach, depending on what is appropriate for each surface.
One of the final steps of our cleaning process is cleaning our waders. We spray our waders down with a Virkon solution, and that will kill any pathogens or any sort of bacteria or anything bad that occurs on our waders during the day when we're in the field. If we switch field sites day by day, it's very important that we make sure we spray everything down so that way we're not spreading pathogens from different populations to another population. Currently, you can see me trying to shake the water out of my boot before I spray them down because there was still some in there from before. But yeah, you just give everything a nice little spritz. Make sure to get both sides of the waders as well as the shoes. And now we put away all of our samples. So we have a separate freezer that's large enough to fit many sample boxes. So currently Kayla is pulling out our white sample boxes from our one field location. Many of our boxes are separated not only by what type of samples they contain, but where the sample was taken from. So here she has the entire stack of white boxes from our one field site that we were at today, and our baggie to put our old plasma tubes in. You can now see we have separate boxes for our CBC blood tubes, our oral cloacal swabs, our shell swabs, as well as our plasma tubes. So lab work from this perspective on the vlog seems pretty simple, pretty straightforward, go from one thing to another, but in all actuality, it kind of looks crazy because Kayla and I are jumping back and forth from one thing to another and often multitasking, leaving us to look something like this. Crazy lab rats running around in circles until all of our samples are done.